Hello, in this video we'll discuss carbohydrate metabolism, particularly glycolysis. Glycolysis is derived from Greek word glycose, which means sugar or sweet, and lysis, which means dissolution or degradation. The term glycolysis means breakdown of glucose to pyruvate or lactate via a series of enzymes. It means the glycolysis in glycolysis there is breakdown of 6 carbon containing glucose into 3 carbon containing pyruvate or lactate. This pathway is also referred to as Embden-Merop-Paranas pathway, that is EMP pathway. Glycolysis is followed by Krebs cycle in the stages of cellular metabolism. Next, that is silent features of glycolysis. This is the major pathway for utilization of glucose, and it is takes place in all cells of the body. These enzymes which are essential for this metabolism of glucose is present in cytosol. That's why it is takes place in cytosol only. And it is also occur in aerobic and anaerobic conditions. It is a major pathway for ATP synthesis in the tissues lacking mitochondria, for example, RBC, lens, cornea, etc. It is very essential for the brain. Glycolysis can be divided into three distinct phases. First, energy investment phase or priming stage. Second, splitting phase. And third, energy generation phase. This is the pathway of glycolysis, that is breakdown of glucose 6 carbon containing compound into pyruvate, that is 3 carbon containing compound. In the first step, glucose is converted into glucose 6 phosphate with the help of enzyme hexokinase or glucokinase. The difference between hexokinase and glucokinase is that hexokinase is present in all the tissues except liver, while glucokinase is present in liver, and this process required ATP to produce glucose 6 phosphate. In the second step, glucose 6 phosphate is converted to fructose 6 phosphate, and this is isomerization reaction. Enzyme catalyzed for this reaction, that is phosphatase isomerase. In the third step, fructose 6 phosphate is converted to fructose 1,6 bisphosphate with the help of enzyme phosphofructokinase, and in this third step, second ATP is utilized. Up to these stages, three stages, conversion of glucose to fructose 1,6 bisphosphate. This is the energy investment step. In the next step, there is a splitting. 6 carbon containing compound is converted into 3 carbon containing two compounds, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are interconverted with the help of enzyme triose phosphate isomerase. It means in the process of breakdown of glucose, two molecules of glyceraldehyde 3-phosphate will synthesize. Then next step, glyceraldehyde 3-phosphate is converted into 1,3-phosphoglycerate with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, NAD is utilized to produce that molecule and NADH is synthesized. In the next step, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, and this is the substrate level phosphorylation step in which ATP is synthesized without involvement of electron transport chain, and this reaction is catalyzed by phosphoglycerate kinase. Next step, 3-phosphoglycerate is converted to 2-phosphoglycerate, and the enzyme is phosphoglycerate mutase. In this reaction, the phosphate group on third carbon is shifted to phosphate group on second carbon. In the next step, 2-phosphoglycerate is converted into phosphoenol pyruvate with the help of enzyme enolase, and the last step is phosphoenol pyruvate is converted to pyruvate with the help of enzyme pyruvate kinase. This is the second step, substrate level phosphorylation reaction, where ATP is synthesized. So up to this stage, from glucose to pyruvate, there is the aerobic uh, aerobic glycolysis. In case of anaerobic glycolysis, that is conversion of glucose to lactate, all the steps are same except last step, that is pyruvate is converted into lactate and the enzyme is lactate dehydrogenase. In this reaction, NAD is synthesized from NADH, and that NAD is utilized in the fourth step, that is conversion of glyceraldehyde 3-phosphate to 1,3-phosphoglycerate. Next, regulation of glycolysis. That glycolysis is controlled by three enzymes: hexokinase, phosphofructokinase, and pyruvate kinase. All these enzymes catalyzes irreversible reactions. Next, that is calculation of ATP produced per glucose molecule in glycolysis. Three main enzymes are there which is used for the synthesis of that ATP: glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, and pyruvate kinase, particularly in step number 6, 7, and 10. In the 6th step, NADH is produced, and that NADH is equivalent to 3 ATP. It means 3 multiplied by 2, that is 6 ATP is synthesized in 6th step. In step number 7, 2 ATP will synthesize, while in step number 10, 2 ATP will produce. It means total 6 plus 2 plus 2, total 10 ATP will produce in that process. While 2 ATP are utilized in the first step, energy investment step, in the first step and third step. So total net gain, ATP produced, that is 10, ATP utilized, that is 2, net gain is 8 ATP. So total 8 ATP are produced in aerobic glycolysis, while in anaerobic glycolysis, the last step, NADH are utilized. It means total 6 ATPs are utilized to convert that pyruvate into lactate. So net gain of ATP in anaerobic glycolysis is 8 minus 6, that is 2 ATPs are produced in aerobic glycolysis. So up to this, it is about glycolysis process. In the next video, we will discuss Krebs cycle and electron transport chain. Keep watching. Thank you.